How can squid ink be used to combat the growth of cancer tumors? I'm Lovely Morning Signey.
I am Cyna Jeanelle, Agra.
I am Phoebe Emmanuelle, Amatorio, and we are from Philippine Science High School Main Campus under SDG 3 Good Health and Well-Being category.
In 2018, there were about 9.6 million cancer-related deaths worldwide. A quick recap on cancer biology. Cancer relies on the process called angiogenesis. Angiogenesis is the growth of new blood vessels. Angiogenesis inhibitors, on the other hand, are substances that stop these vessels from growing new ones, which cuts off the blood supply that the tumor needs in order to grow. Anti-angiogenesis is currently considered as a possible solution to combat the growth of cancer tumors. That said, the ink of cephalopods, a class in the phylum Mollusca, has been reported to contain substances that inhibit angiogenesis. We plan to investigate if the species Sepioteuthis lessoniana, one of the most common squids in the Philippines, inhibit angiogenesis. But how?
The Chorioallantoic Membrane assay or the CAM assay will be used as the model for angiogenesis. When a substance, in our case our ink, is introduced to the CAM, we'll be able to identify its anti-angiogenic effect on the vascular of the CAM. So, our objective is to determine whether or not the ink of Sepioteuthis lessoniana inhibits the growth of new blood vessels in the CAM assay and thereby possesses anti-angiogenic effects.
Samples of the squid were procured and underwent species verification. 200 ml of ink was freeze-dried into 66.5 grams of powder. 50 8-day-old fertilized eggs were also procured and viewed through an egg candler to mark the air bubble where a 1 by 1 cm window was drilled. Simultaneously, 5 treatments were prepared. Different concentrations of squid ink in Phosphate Buffered Saline or PBS, a neutral solvent, and Quercetin, an effective angiogenesis inhibitor. There were 10 replicates per treatment, so 10 filter paper discs were soaked in each of the treatments and applied to the drilled hole of the egg and then sealed again with parafilm and incubated. After 48 hours of incubation, the eggs were harvested and placed into Petri dishes where the CAM was viewed and documented. ImageJ software was used to quantify the number of blood vessels in the CAM assay after the treatments were applied.
The results were tabulated in this way. As expected, the negative control had the most number of branching points in all, because no treatment was applied, while the positive control had the least number of branches, as quercetin is known to be a good angiogenesis inhibitor. Statistical analysis showed that the ink treatments were significantly different from the negative control and had no significant difference with the positive control. Lyophilized ink from S. lessoniana inhibited blood vessel formation as effective as the known anti-angiogenic inhibitor quercetin.
Anti-angiogenic effects of the ink can be attributed to the presence of sepia ink polysaccharide or SIP, since some species of squid and cuttlefish have been identified to have SIP, although SIP still hasn't been isolated from this particular species. These polysaccharides have unique molecular characteristics, structure, properties, and possess activities.
We can conclude that the ink of S. lessoniana successfully inhibited angiogenesis in the CAM and thus contains anti-angiogenic properties. It can also be concluded that the anti-angiogenic effects of the squid ink is not dose-dependent at 6.25% to 25% concentration, although further studies needed to confirm whether SIP is present in the squid ink of the species S. lessoniana. Further studies also needed to quantify at which exact concentrations the anti-angiogenic effect is dose-dependent.
This finding can be used to treat diseases like cancer. It can be developed to become a drug or medicine for patients.
Furthermore, it offers an alternative to current mechanisms in cancer therapy, and it holds promise as a drug to manage other angiogenic-related diseases.